Chapter. It's not a chapter, it's a part of chapter. It's a very simple chapter. It's a conceptual chapter. So let's start to understand it. Let's start to understand the concept of this chapter. So chronic inflammation, right? What is chronic inflammation? In simple words, chronic inflammation is lymphocytes plus what is chronic inflammation? Chronic inflammation is simply lymphocytes plus plasma cells. That is chronic inflammation. Okay. So if there is presence of lymphocytes and if there is presence of plasma cells, that is chronic inflammation. Okay. Now what starts it? Well, sometimes it's not directly after acute inflammation. It can be benign. It can begin as a chronic form from the start, like autoimmune diseases. And why it happens? It happens because T cells and B cells are involved in it. Okay. Because T cells and B cells, they take longer to get recruited, and neutrophils are faster. So T cells and B cells, they take longer to get recruited, and neutrophils are faster. So that's why neutrophil rich is acute, and lymphocytes and plasma cells mediated inflammation is chronic inflammation. Okay, let's start the story. Let's start the movie of chronic inflammation. So what is chronic inflammation? Inflammation bringing out immune cells into tissue. Chronic inflammation mainly lymphocytes, I already told you. In acute, it's mainly through neutrophils, and chronic is delayed and it's adaptive, right? It's more specific. It's not innate. Acute inflammation is innate immunity, but chronic is delayed. Okay. What are the stimuli? We know the stimuli. It can be persistent infection, can be persistent virus infection with virus, mycobacterium, parasite, fungi, foreign material, some cancer, autoimmune disease. It can be a lot of things. Okay, let's start with T cells. Okay, so T cells explain the development and maturity of T cells. So we have something called as T cells, right? What are T cells? T cells are cells that are made in bone marrow, and they go to thymus for education. Okay, so thymus, they become specific to antigen. Thymus is like a college, school. So thymus teaches T cell what to attack, what not to attack. Okay, so thymus, they become specific to an antigen. Thymus is the place where they get educated about an antigen. It's the learning place, right? And they develop into either CD4 helper and CD8 toxic. So CD4 helper, it means it's helping in something, and CD8 toxic, it means it's cytotoxic, it's gonna kill. That's it. So you can either become CD8, CD4 in life, right? You can become a helper or you can become a killer. Okay, so how CD4 and CD8 gets activated? Well, CD4 and CD8, they get activated via different pathways, right? So what is their second message for activation? Well, to get activation for CD4 and CD8, you need, first of all, their own self, right? Their own CD4 cells, and you need a second signal as well. Now, why do you guys think that? Why do CD4 and CD8 cells need second message signal? Well, they need second message signal because you don't want to get autoimmunity disorders, right? Imagine if you have something wrong, right? Your body starts to make autoantibodies for yourself. Imagine if this happens. You must have some sort of handbrakes on your autoimmune systems. So this handbrake is the second system message. If this second system message is not, messed up, then you guys will not have any sort of autoimmune disorders. But if you guys have this second system message messed up, it means this autoimmune disorders can start to occur, right? So we need, first of all, themselves, CD4 cells and CD8 cells for activation, plus we need the second messaging signals, right? These are like permission granted to change, permission granted to develop. So that's the concept of second message signal. That's why we need them. So let's talk about it. What happens is that T cells are activated. Now, when they recognize antigen presented on MHC molecule, what is MHC? What is MHC? This is MHC, major histamine compatibility receptors. Complex. Okay, so it's a complex of histamine, right? Okay, so basically MHC is like a showcase, right? It's a transparent glass window, and it's a transparent glass window for CD4 cells and CD8 cells. Okay, so what is the main concept? We'll read all of this. Okay, we'll read about all of this later. Let's start to study my my summary, right? Let's start to understand chronic inflammation simplified, right? I wrote this. Okay, so let's start. Why second signal needed? I told you it's a double check for your autoimmunity danger. If 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 any CD8 is highly reactive to itself, if it's self-reactive, then it undergoes apoptosis, right? It's a physiological way to die, right? No inflammation after apoptosis. So that's why you need second signal. It's like a guarantee for no autoimmune dangers. Okay. What is MHC? MHC is major histamine compatibility complex. It's like a display piece for your antigen to T cells. Okay. Thymus, where is thymus? Thymus is present. It's like a school for T cells. T cells go to thymus, and then T cells get educated, and then later on T cells are the teachers itself. Okay, so that's the concept. What is TH? Here you see TH is T helper. That's it. Okay, so B cells have 5 isotype. What is 5 isotype? It means they have 5 same chemical structure, but the physical structure is different, right? Same chemical molecules, number of molecules same, everything same, but the structure is different, right? So IgM, it's just they have same FAB region, and they have different FC region, right? So a naive B cells makes IgM and IgD. So naive means newly formed. Okay, so a newly formed uneducated guy who has not yet got the education from T cells, it only makes IgM and IgD. So the whole mess, the whole chronic inflammation, the whole story is about conversion of these B cell from IgM to IgD into IgG, IgA, and IgE. So if there is any problem in this whole process, it means you'll not be able to make IgG, IgA, and IgE antibodies. Okay, so that's clear. So the whole mess is about how a naive B cell that only makes IgM, with the help of T cell, now a teacher, makes IgG, IgA, and IgE. Okay. So another concept to understand is that when we change these classes, it's called, when we change these classes, we call this class switching. Okay, we call this class switching. We keep the same antigen specificity, but we change the backend. We call it class switching, right? So CD4TH, it's the college of B cells and macrophages. So I just told you these are B cells, right? These are immature plasma cells, right? And macrophages, these are immature monocytes, right? So if you have problem in these CD4 or CD8, then your teachers are messed up. If you have problem at thymus, it means the school of those teachers are messed up. So you gotta have good school, you gotta have good teachers, so that you can have good IgM, IgD, IgE, IgA, right? Antibodies. So this is the whole story about, right? Another thing is that CD4 cells and CD8 cells don't go to other cells. Other cells come to them. Okay, these are the teachers. Okay, you go to teachers. Teachers don't come to you, right? Okay, so this is very important. So whenever they want any sort of education, for example, B cells want education, macrophages want some education, they go to CD4 cells, right? And how they present their cases, how they present their discussion, how will you take it up to your seniors? You will write an application, right? Like, hey, this is going on, I'm getting bullied, please help me, blah blah blah. So this application is MHC, this showcase of antigen. Like, hey, like the B cell goes to CD4 cells and says, hey, like, I just got this rabbit. Are you sure this rabbit is not harmful? So how will this B cell show this rabbit to CD4 cells. How? Via MHC2 and via MHC1. Now MHC2 is the showcase for CD4 cells, okay? And MHC1 showcase, MHC1 glass, MHC1 plate, right, is the showcase for CD8 cell. So 4 into 2 is 8 and 8 into 1 is 8 also. So that's how I remember, right? So MHC2 class is for CD4, MHC class 1 is for CD8, and CD8 is cytotoxic and CD4 is educative, helper. Okay, so this is the concept. Now I'm building a concept. Let's start with the, so chronic inflammation means lymphocytes plus plasma cells, right? T cells mediated, it's all T cell mediated, right? T cells are the teacher cells. And so T cells, they come from thymus. Thymus is the college. Now, after thymus, they get separated into CD4 helper cells and CD8 helper cells, right? CD4 helper is helper, CD8 is killer. It's just kills the cell, okay? So to get activated, CD4 helper cells, I told you, they need something. They need a second message signal and they need something antigen, right? So what happens that CD4 helper cells get activated with MHC2, right? Antigen complex, okay? Let's read the line. So helpers, okay. So T cells, both CD4 and CD8, get activated when they recognize antigen presenting, something is presented on MHC molecule, okay? Okay. And the second signal is also present. So you have to have MHC presenting antigen and second signal, both. If you are missing second signal, you won't get activated. So CD4 T cells read antigen from MHC2, right? And they are extracellular antigen, means outside, okay? It's not internal environment, it's outside environment, okay? And are present on antigen presenting cell, right? Antigen presenting cell, right? Whereas cytotoxic CD8 read antigen from MHC1 and present intracellularly or viral antigen that are present in the cell, right? So all the cells that have nucleus, they have these MHC1 molecules. And this MHC1 molecule is the showcase. It captures the antigen from within intracellular environment, right? So this MHC molecule with antigen goes to CD8, plus there is some second message, there's some second signal, a permission to change, right? And then they get activated. So I hope you get me. So CD4 gets activated with MHC2, plus there is an antigen, plus there is a second signal, okay? And CD4 killer get activated when there is MHC1 molecule that is presenting an antigen from internal space, plus there is a second signal, okay? So you guys need a second signal plus MHC1 for CD8, and for CD4 you guys need MHC2 antigen and a second signal, okay? So you guys need to have an antigen, MHC and second signal. So these three things you need for activation, okay? So second signal for CD4 is different, second signal for CD8 is different. Let's talk about CD4 first and then we go to CD8, okay? So what happens is that CD4 gets activated with second signal, right? And MHC has an antigen from extracellular space, right? So what's the second signal for CD4? The second signal for CD4 is B7 molecule, okay? It's the second signal. So B7 plus CD28 on CD4 cells. So CD4 cells, these helper cells have a CD28 molecule on them here. And this CD28 gets attached to B7 molecule, okay? So once B7, it attaches to CD28 receptors on CD4, it's get activated. So we need this thing, B7, 28 and CD4. So when you divide 28 by 7, you get 4, right? So this is the second signal molecule, B7 molecule, and it only attaches to CD28, which is already attached on CD4 molecule. So I hope you understand this. After this, the activation happens. Now, as soon as the activation happens, CD4 cells can differentiate into TH1 and TH2, okay? TH is T helper 1, T helper 2, right? So T helper 1, so 1 is priority, right? So priority is defense, right? So priority is defense. So what is defense? Macrophages, right? They do opsonization, they do phagocytosis, right? They're bactericidal cells, right? They kill the bacteria. So Th1 cells, they produce something called as interferon gamma. Now, if I'm saying gamma, it means there are some interferon beta. It means there are some interferon alpha. Yes. So interferon gamma. So whenever I see gamma, it means green. Gamma stands for green in my mind, because I saw these movies growing up, Marvel, and I saw that a guy got exposed to gamma radiation and then he became Hulk. So this macrophage is like a Hulk in the body, right? So Th1 makes interferon gamma, which is a green thing, right? Now you can see it's a green, and then they make macrophages. So now one pathway is clear. Okay, Th1 makes interferon gamma, makes macrophages. Okay, after activation of CD4, we make Th1. We already know. Now we make Th2 as well. Now this Th2 is other thing. It's the pathway. It's the main reason for this whole mess, the whole story. What happens is that Th2 makes something called as interleukin 4, interleukin 5. Now these interleukin 4 and 5 are in same class. Why I'm saying they are in same class? Because they more or less do the same thing. Both interleukin 4 and both interleukin 5, they teach B cells to change class switches, okay? To change the classes, okay? Because B cells, when they are naive, when they are kids, they only make something called as IgM and IgD, okay? So they need to make these immunoglobulin complex IgG, IgA and IgE. They need it, but they cannot because they are stupid. They are young, right? They need education. And who's gonna teach them? Th2 is gonna teach them, okay? So Th2 teaches B cell and just B cell to convert from these stupid form it's not stupid, these forms from IgM to IgD, and switch the class from IgM to IgD into IgG, IgA and IgE. So we need to switch the classes. So what happens is that IL4 changes the class to IgG and IgE, to IgG and IgE, okay? And IL5, interleukin 5, tells the B cell, the plasma cell, to mature into plasma cells, right? Okay, it's IL5 tells, okay, grow up, guys, you need to change, and it changes the class to IgA, So IL4 changes the class to IgG and IgE. So these are 4, and IL5 changes to IgA, okay? And the maturation of plasma B cells, okay? Now we understand this whole concept, whole mess. What about the other part, IL-10? Now I'm saying IL-10 because I made this in red color because it's an opposite. It is anti-inflammatory, okay? It is anti, it's not hentai, it's anti. So anti-inflammatory, it means it's opposite. It wants to stop whole this thing. Now I told you, if you stop the Hulk, you stop everything. So what IL-10 does is that it secretes anti-inflammatory cytokinins, and then they inhibit Th1, T helper 1, so that no Hulks are produced, okay? So IL-10 inhibits Th1, IL-4 produces IgG, IgE, IL-5 produces IgA classes, okay? So now you understand this concept. What happens afterwards is that macrophages, the Hulk of the body, they too have, MHC molecules. They too have MHC, glass pieces to show antigens. Now what happens that macrophages go to CD4 again, and they present the antigen via MHC2 molecule, because these MHC2 molecule can handle antigen from extracellular space, okay? But MHC1 molecule cannot handle the antigen from extracellular. So MHC1, class 1 molecule, handles the antigen from intracellular space. So what happens that macrophages, they go to CD4 helper cells and they tell, okay, hey, look at this antigen I got from outside via MHC2. And then CD4 cell is like, okay, let's do something about this antigen which you are showing. Let's try to, give you a new form of inflammation. So what happens is that this they show. So after showing it to CD4 cells, what happens that CD4 starts a new system. What is the new system? The whole this system stays the same, where CD4 makes activation then makes TH1 then makes gamma, and then gamma, instead of going into macrophages, it changes into granulomatous inflammation. So granules, okay, granules, okay, so granules of inflammation, right? And then this granular inflammation can be of two types, caseous and non-caseous. Caseous is cheesy and non-cheesy, okay? So this is a whole story about this macrophages, granulomatous inflammation, caseous, non-caseous, okay? And how IL-4 makes IgG and IgE, okay? This is the whole story. Now, another part to the whole story is B cells, right? B cells are these stupid kids, right? They want education from T cells. So what happens is that they also want to, change switches. They also act independently. They also want to feel important. So what happens is that they also need something called as second signaling. Because imagine if you have autoimmune disease for B cells. If you have messed up B cells, then you'll have messed up IgG, IgE cells. So they need a second signal as well. So the second signal for B cells comes from a molecule called CD40L. Now this CD40L means ligand. Ligand is something that binds to the receptor. Okay. So B cells have a receptor called CD40 receptor. And CD4 cells, the teacher cells, these teacher cells have these ligand called CD40L, and B cells have these receptor CD40. So they do a binding, they bind with each other, and then this second signal is born. Now what this second signal says? This second signal says the CD4 cells tells to B cells that, hey B cell, you have the permission to switch classes, you have the permission to mature, you have the permission to do whatever you want. Now the B cells can, grow up. Now the B cells can go to another city. They can, invade other things. Now the B cells can grow up. So this is the whole story about how CD40L is the second signal, right? So B7 is the second signal for CD4. IL-2, okay, let's talk about CD8 killer. I completely forgot about CD8 killer. Okay, so CD8 killer, they are killer. It means they kill. So CD8 killer needs MHC1, that is the showcase molecule presenting antigen, right? And it presents antigen from intercellularly because it's a small plate, okay? So it cannot handle antigens, big antigens from extracellular space. It needs smaller antigens, right? So it needs MHC1 molecule plus antigen plus a second signal. Now what is the second signal for CD8 killer? IL-2, okay? So IL-2, where does this IL-2 comes from? Now this IL-2 comes from TH1, because TH1 makes interferon gamma and IL-2, okay? So TH1 makes IL-2 and interferon gamma. TH2 makes IL-4, IL-5, IL-10. IL-4 makes GE antibodies. IL-5 makes IgA antibodies. And IL-10 is anti-inflammatory, inhibits TH1. So if we have more number of IL-10, so it will inhibit TH1. Once TH1 is inhibited, it means you guys have less number of IL-2. If we have less number of IL-2, the CD8 will not get activated. If they do not get activated, they will not perform their function of phagocytosis, bactericidal, opsonization. They will not do these functions. So IL-2 plus MHC1 plus the antigen presenting MHC1, okay, it results in activation. So this was the whole concept of chronic inflammation. It's chronic because it takes time, right? It needs lymphocytes. And this whole mess is about making sure that you guys have IgM, IgD. IgD is immature B cells. IgG, IgA, IgE, these are class switching from via IL-4, IL-5, IL-4, IL-5, right? And what else? And CD4 cell, we have to mature these cells as well. We have to tell them, okay, gets activated. How they will get activated? They'll get activated by TH1. TH1 get activated via CD4. So this is the whole story about chronic inflammation. Now we can read the text, understand the text. I hope, okay, let's see, read the text. Okay. So what is the stimuli for chronic inflammation? We know this, cancer, blah, blah, blah. T cells, they go thymus. Okay, we know this. Let's read this. Okay, so CD4, CD8, second message activation. So CD4 T cells read antigen from MHC class 2. MHC class 2 present extracellular antigen, and they are present on antigen present cells, right? They are extracellular. It means they have big molecules, right? And cytotoxic CD8, they read antigen from MHC class 1. They present intracellularly or viral antigens. They present on all cells of the body, right? So all cells, it means all cells that have a nuclei in it. If a cell doesn't have a nuclei, it means it cannot present MHC1 molecule. Okay. So for CD4 cells, B cell is the second messaging signal, right? That's bind to CD28 on CD4 cells. I just showed you. For CD40, for B cells, CD40, so binding of CD40 and binding of CD40L is the second signal. Okay. And CD8 cell, IL2 IL2 from CD4 cells, I just told you, IL2 is made from TH1. And TH2 makes IL4 and 5 and 10. IL4 and 5 for Ig bodies and IL10 for anti-inflammatory. TH1 make gamma and IL2. Okay. So CD4 makes IL2. CD4 cells second messaging for activation. What do activated CD4 cell do? Again, these are helper cells. They secrete cytokines. They have two types, TH1, TH2. Now TH1, I told you, makes interferon gamma and IL2. Now IL2 activates CD8 and interferon gamma makes macrophages. We know this. What about, explain the purpose and function of cytokines secreted by activated CD4, TH2. Now these are TH2. I told you TH2 makes IL4 and 5 and 10, right? So TH2 makes eosinophilic rich inflammation. Eosinophilic rich inflammation means acute inflammation. And they reduce inflammation. How? By secreting IL10, right? And how they make this inflammation? By making IL4 and 5. So IL4 and 5 for rich inflammation and IL10 for inhibiting inflammation, reducing inflammation. So IL4 causes class switching to IgG and IgE. IL5 attracts eosinophils, helps maturation of B cells into plasma cells, and causes class switching to IgA. So I told you IL4 and 5 makes IG, which one? IgG and E. Okay. IgM and D are naive B cells. G and E, IG4, sorry, the EL4, the IL4, the and IL5 is for IgA. Okay. Okay, this is, we know this. Okay, we know this. Inhibition, TH1 phenotype, we know this. So how do CD8 killer cells, right? How do T cells kill cells? How do they do it? They secrete something called a performance. Perforance means they perforate the cell wall of the enzyme. This is not the enzyme. Perforate the cell wall of the host cell. They make holes in the host cell, secrete something called as granzymes. Now these granzymes activate something called a caspase. Now we need caspase for apoptosis, right? Because caspase, what caspase does is it reduces it renders cytochrome C inactive. And cytochrome C, we need cytochrome C. Cytochrome C is the guard for this apoptosis signaling, right? So it induces caspase system and apoptosis happen. Other thing that happens is this fast ligand, fast receptor. Now okay, there are three ways CD8 kills the cell. Let's just revise it. Molecular weight, wait, because it's very simple chapter. Wait, where it is. Cytochrome C, phagocytosis. There it is. So what are the three pathways for caspase activation? Okay, so caspase can induce this apoptosis in three ways: intrinsic inside, extrinsic outside, and cytotoxic CD8. Let's talk about cytotoxic. So CD8 makes perforins, releases granzymes, enters target cell, and does this apoptosis happens. Intrinsic is that there is something called as BCL2 molecule. Now this BCL2 molecule, this BCL2 molecule is the guard for cytochrome C molecule, right? This is mitochondrial membrane. What happens that inside the mitochondrial membrane there is cytochrome C molecule, and then this molecule is present there, right? So if this molecule gets out of this mitochondrial membrane, what happens is that apoptosis starts because this is not there inside. So BCL2 is the guard that keeps the cytochrome C inside. Once the BCL2 is rendered ineffective, what happens that cytochrome C is active, and then caspase is active, and then apoptosis happens, right? BCL2 is a protein. It stabilizes mitochondrial membrane. Cytochrome mitochondria can't leak out. If the membrane damages, DNA damage, decreased hormonal stimulation, lack of estrogen in your period cycle, the BCL2 gets inactivated, leakage of cytochrome C, activation of caspase system happens. Extrinsic pathway, again we read about fast ligand binding. What happens to this fast ligand? This thing is that, so this is a fast, right? Okay, wait. Fast ligand binds to fast death receptor CD95. So on a cell there are this receptor called CD95, and this is a fast. And okay, so once this fast binds to CD95 receptors, apoptosis happens, right? This is simply in simple words. So what happens is that the school, right? Let's read it. So new CD T killer cells from bone marrow undergo positive selection and negative selection. So in positive selection, what happens is that if the CD8 cell bind to self antigen on MHC1 molecule, they get to live, right? Okay. But in negative selection, what happens is that if the T cell, if the CD8 cell bind to self antigen too strongly, like they have this tendency to attack on itself, in thymus, what happens that they get killed, they get killed, right? They undergo apoptosis. So this negative selection via fast ligand binding to fast death receptor CD95, they die. Another example is TNF, tumor necrotic factor, binds to tumor necrotic factor receptor, and once TNF binds to TNF receptor, apoptosis happens. So these are three ways: intrinsic, extrinsic, and cytotoxic, right? Okay, let's see. Okay, we know this fast ligand, we know this. Okay, so how is self-reactive T cell killed? If there is self-reactive T cell, it will bind to MCH antigen, but the second signal won't be present. So that's why we need second signal. It's like a guarantee of not self-reacting T cell. So this is apoptosis or energy. Okay, what is B cell? How B cells are produced? Well, immature B cells are produced in bone marrow. They are called naive B cells. They express IgM and IgD. Our work of this whole mess of CD4 T cells and CD8 T cells is to make sure that they switch classes between IgM and IgD to IgG, IgE, and IgA. IgA is done by interleukin 5. IgE and IgG is done via interleukin 4, right? It's very simple. So how are two ways by which B cells activated? Isotype switching occurs. Well, binding of antigen, they need to bind to an antigen, right? And then these binded antigen B cell, they get activated, right? And how will they present? If they are presenting to CD4, they will present by MHC2. If they are presenting CD8, they will present by MHC1. Okay. So what happens is that they need CD40L plus CD40. It binds, and then we have something called a second signal for activation. So Th2 cells, they secrete IL4, IL5 for, that IL4 we need for IgG and IgA or E, and IL5 for the remaining Ig. So this is how the plasma cell maturate via IL4, IL5. What is granulomatous? What is granulomatous inflammation? Well, it's a chronic inflammation. The number one characteristic of this inflammation is epithelioid histocytes. Epithelioid histocytes means on the epithelial there are some histocytes, histocells. There are some cells. What is this cell? This is compact macrophage. What happens? Macrophage with abundant pink cytosol. Usually, what happens is that macrophage have something called as foamy cytosol. It's very, dispersed. It's not very concentrated. Now, once these cells are made, epithelioid histocells, these are macrophage with dense, abundant pink cytosols, right? And elongated nuclei, very high yield. You may also see multinucleate macrophages in healthy tissue. So macrophage normally have something called as clear foamy cytoplasm. Once they undergo this granulomatous inflammation, they change, dysplasia happen, not dysplasia, they change into this abundant pink cytosol. So granulomas are surrounded by a rim of lymphocytes. So this is granuloma, this is lymphocyte. Okay, so lymphocytes and giant cells, they are giant. Only a certain condition give granulomas, so we can identify this disease. So if you see granulomas, we can be sure of some disease like foreign material. For example, leaking of breast implants. They can cause formation of granulomas. Sarcoidosis. They can also a hallmark of granulomas. Beryllium exposure. It's also one of the hallmark of granulomas. Hallmark of ulcerative colitis is crypt abscess. Okay, and cat scratch disease on your neck, gives star-shaped granuloma. So if you see granuloma, think about these diseases: sarcoidosis, beryllium exposure, Crohn disease, cat scratch, foreign material. Okay, and granulomas can be divided into caseating or non-caseating. Okay, so let's talk about non-caseating. What causes non-caseating? Definition: feature of non-caseating granuloma is lack of central necrosis. So in the center, if there is no necrosis happening, if the center is okay, they are non-caseating, they are non-cheesy. If the center is messed up, if the center has necrosis, they are cheesy. They are cheesy necrosis. They are cheesy granuloma. What causes caseating granuloma? Number one part, TB and fungus infection. Now this caseating necrosis also happens in TB and fungal infection. What caseating is that? This looks like a pasta sauce. Okay, it's thick as a pasta sauce, and this pasta sauce is because of infection from bacteria, right? So we need TB and fungal infection for caseating inflammation or caseating necrosis. So characteristic of TB fungal infection, you see caseating inflammation. The test for these granuloma is first AFB stain for TB to look for TB, and other is GMS silver stain to look for fungal infection, right? Okay, how do granulomas form? Well, I told you macrophage present antigen CD4 to MHC2 extracellularly. Because it's, are you sure it's extracellularly or intercellularly? Yes, I am sure. After binding macrophages, they secrete IL-12. That induces IL-12. So after binding to macrophages, macrophages also secrete. So macrophages make IL-12 that induces CD4 cell to differentiate into Th1 subtype. So Th1 subtype now is done via IL-12, right? So let's see. Let's correct this graph a little bit. Not graph, this drawing. So what happens that here the showing is happening, right, via MHC2 molecule. Showing is done, right? So this via IL. Okay, so Th, what happens that, okay, macrophage, they make something called as IL-12, interleukin 12, right? And this interleukin 12 makes Th1. Okay, and then Th1 makes this interferon gamma, and then this gamma leads to granulomatous inflammation. Okay, so IL-12 induces CAD4 to differentiate into Th1 subtype. Now this Th1 subtype secretes gamma, interferon gamma, converts macrophages into epithelioid histiocytes, and this epithelioid histiocytes and giant cytes. Epithelioid histiocyte means dense pink cytosol. Okay, usually macrophages have foamy transparent cytosol. Okay, so granuloma hence is formed. Both of this, these steps occur in both caseating and non-caseating granuloma. So that's chronic inflammation right there. I hope you understand the whole concept of this, because this concept is very important. Once you understand this concept, it's a very simple concept. I hope you understand this whole concept. Have a nice day. Okay, how to stop this recording? Okay.